As we begin our study of microbiology, I think it's good to go back and see sort of how the science of microbiology developed. Microbiology, if you break it down in terms of medical terminology, ology means the study of, micro means small, and bio means life or living. So microbiology is the study of small life forms or small living things. We refer to these as microorganisms or microbes. Most microbes are not harmful. In this class, we are going to focus on the pathogenic ones, the harmful ones, but most microbes are actually beneficial to us. When something lives at the expense of another organism, we refer to it as a parasite. Parasites are going to damage the host organism, the organism on which it's living. Pathogens are organisms that cause disease. Parasites and pathogens are somewhat interchangeable, but the vast majority of microorganisms, 90 or more % of microorganisms, are beneficial to us, or at least don't bother us at all. They make food, they're used in bioremediation, they make antibiotics, just to mention a few of the beneficial things that microbes do for us. Microbiology covers a lot of different kinds of organisms. We will talk about bacteria. They'll kind of be the focus of this class. But we'll also look at fungi, algae, protozoa, and helminths. Helminths are worms. And we will also look at viruses. When microbes interact with humans, you have to remember that microorganisms are ubiquitous, which is just a big word that means they're absolutely everywhere. Microorganisms are involved in what we call nutrient and energy flow. Some microorganisms are capable of photosynthesis, so they take raw materials and turn it into more complicated materials that other organisms can use. Microorganisms are also involved in decomposition. They take those big biomolecules and break them down into smaller molecules. Basically, that armadillo on the side of the road eventually disappears, partially because of microbial action. Most microorganisms are beneficial: food, antibiotics, and bioremediation. To say nothing of our normal flora. You have more microorganisms living in you and on you than you have cells in your body. And your normal flora either provides you with something you need, like the E. coli in our gut provides us with most of our vitamin K, or they may simply take up space in surfaces and prevent other harmful organisms from living there. At the very worst, they're just living there and they don't harm us. But a very small percentage of microorganisms are pathogenic and do cause disease. And unfortunately, we will be focusing on the bad guys in this class. There are different types of microorganisms. Most of the bacteria, well, all of the bacteria, most of the things we will talk about are prokaryotic cells. Now, prokaryotic cells are very simple cells. They don't have a membrane-bound nucleus. They have no membranous organelles. They're pretty simple organisms. They're fairly small. And then we will study some eukaryotic cells. Eukaryotic cells are the more complex cells. They have a membrane-bound nucleus. They have a lot of organelles like mitochondria and Golgi apparatus. If you have taken any other biology courses and talked about cells, you have probably talked about eukaryotic cells. And then finally, we will look at viruses. Viruses are not cellular. They're said to be acellular. They are really particles. They are composed of a protein coat and one nucleic acid. They contain either DNA or RNA, but they never contain both. In the very early years, the ancient Greeks knew a little bit about microorganisms. They talked a little bit about them. They thought there were invisible things that were somehow involved maybe in making us sick. But it wasn't until the late 1600s that Anton von Leeuwenhoek actually got credit for sort of starting microbiology. He developed the first microscope. Leeuwenhoek was not a scientist. He was a tailor and bought linen as part of his tailoring business. Because linen was woven, he wanted to make sure he was getting very high quality linen. And sort of as a hobby, he ground magnifying lenses. So he developed fairly high-powered magnifying lenses that would allow him to examine the fabric he was purchasing and make sure that it was high-quality fabric, high-quality linen. But Leeuwenhoek was very curious, and he used his special lens to look at pretty much everything. He looked at pond water. He looked at teeth scrapings. He looked at his own semen. And he did very detailed descriptions of everything he saw and made very detailed drawings of everything. He sent this material off to the Royal Society in London, who made him a member. Now, this was a really big deal in scientific circles in those days, and still is. His drawings are still on display at the Royal Society in London, and people can look at them and actually tell what he was looking at. Now, what's pretty amazing is this is what his microscope looked like. It's not much more than a fancy magnifying glass. It only magnifies a little bit, but he was able to see a great many things in it. For those of you who have ever tried to use a microscope before and have been frustrated with the art of focusing, you are going to love Leeuwenhoek's approach to focusing the microscope. Once he got something in focus on this baby, he just built a new microscope and started over. At his death, there were something like 200 of these microscopes available that his daughter inherited. He never told anyone how he made them. His daughter did not share them with the scientific world for them to try to figure out how he made them. And as a result, at his death in the early 1700s, we kind of stopped looking at microscopic stuff. And microbiology kind of languished for about 200 years until people got interested in microscopes again. A little later, along about the 1700s, a Swedish guy named, actually his name was Carl Linné, got involved with taxonomy. And taxonomy is the science of classification and naming. And he got really carried away with this. He used Latin. Latin's a dead language, so its meaning doesn't change. And he even Latinized his name, so Carolus Linnaeus is how we know him. He was a very modest man. One of his quotes is, "What God creates, Linnaeus classifies." No ego there at all. He came up with a series of boxes that he put stuff in. Basically, he just wanted to bring order to all living things and look at their characteristics and classify them according to their characteristics. Now, first he placed them in a kingdom, and when Linnaeus started, they were either plants or animals. Those were your only two choices. Once he had them in kingdoms, he looked at them a little more closely and placed them in phyla. Those that were a little more alike went into this phylum, and those were a little different but were more alike went into this phylum, and so on. Then he further divided the phyla into classes. Now we're getting more and more specific about the characteristics we look at. Then he went to order, then family, and finally genus and species. As I mentioned, Linnaeus used Latin for naming things because Latin was a dead language. A dead language, the words retain their meaning. They never change their meaning. So he wanted to use Latin words to tell people, some characteristic of the organism. Latin, because it wasn't changing its meaning, was a good thing to use. Let me give you an example of a language that's not dead, English. Today, if we said somebody was gay, it would mean they were homosexual. In 1910, if you said someone was gay, it would mean they were carefree and very happy. The meaning changed over the course of a century simply because the language changes. Latin doesn't do that because no one speaks Latin anymore. Linnaeus came up with what he called binomial nomenclature. That is, binomial, two names for naming something. If we got down to a genus and a species, we would be talking about one very specific organism. The genus name is italicized or underlined, and we use a capital letter when we write the genus name. The species name is likewise italicized or underlined. We always want to set a genus and species off from the other letters or words in the statement, but you do not capitalize the species name. And a lot of you look at genus and species as sort of like a first name and a last name, and you're used to capitalizing a first name and capitalizing a last name, and you've got to break that habit because genus is capitalized, but species is not. We use names that are descriptive. Aureus, Staphylococcus aureus. Aureus means gold, and when that organism grows, it has kind of a golden pigment to it. Or we honor a scientist that discovered it or worked with it. Escherichia coli is named for Dr. Escherick, who discovered it and worked with it heavily. The names have some meaning to them. A genus and a species always denotes one specific kind of organism. Everything in this category are all identical. When you write something like Bacillus cereus, Here's an example of italicizing it, capital genus, lowercase species, or underlining it, again, capital genus, lowercase species. Now I'm going to skip ahead into chapter 4 to talk a little bit more about classification here, because classification is not something that's going to be a big feature of this class, so we'll just kind of get it out of the way here. Linnaeus, as I said, only had 2 kingdoms, Plantae and Animalia. But by the middle of the 20th century, we had done enough division of stuff that there were 5 kingdoms, Plantae, Animalia, fungi, protista, and prokaryote. Originally, we used structural characteristics to identify these things. We also looked at fossil records, and then we started looking at DNA to show relationships and tried to come to some consensus about which organisms belonged where and what family they belonged in and what class they belonged in, and so on and so forth. Bacteria were always a problem, however, because they didn't have a whole lot of structure, and it's really hard to find fossil records of bacteria. Not impossible, but very challenging to find them. A guy named Berge developed Berge's Manual of Determinative Bacteriology, and in it he used very common characteristics of bacteria to identify them. He classified them using gram stain results and then various biochemical tests. We still use a lot of the stuff that's in Berge's manual when we do identification of organisms in the laboratory. In the late 1960s, a scientist named Carolus began looking at a whole new structural chemical to try to determine classification of organisms. Ribosomes are those organelles in the cells that are responsible for protein synthesis. Ribosomes have RNA as a component. Because protein synthesis is a pretty consistent process, that ribosomal RNA does not undergo evolution at a very fast rate. And what Woese started doing is looking at the ribosomal RNA of organisms, and he reclassified things based on their ribosomal RNA. What he discovered is that there were 3 basic types of ribosomes. And he said, let's step back even one level higher than kingdom and put things in domains. And he had 3 domains: eukarya, bacteria, and archaea. Eukarya contains all of the things that are composed of eukaryotic cells. Bacteria are prokaryotic cells, as are the archaea. They're also prokaryotic cells. He's further looked at this, and the ribosomal RNA suggests that in bacteria there may be at least 50 kingdoms, that there are probably at least 3 kingdoms in archaea, and we retained 4 of the kingdoms, Plantae, Animalia, fungi, and protista, in the eukarya domain. Prokaryote is where the bacteria and archaea were lumped prior to Woese's work. Berge's manual, upgraded a little bit in 2010. Berge's Manual of Systematic Bacteriology is now a comparison of bacteria based on their ribosomal RNA. So a few things changed up within the bacterial system as well. Now, as we look a little more at classification, the bacteria and the archaea are composed of prokaryotic cells, and they're single cells. As a reminder, prokaryotic cells do not have a membrane-bound nucleus, and they have no membranous organelles at all. Bacteria have a special carbohydrate in their cell wall called peptidoglycan, and when they look at the cell wall of archaea, they find not peptidoglycan, but some other unique carbohydrate molecules that are not found in any other cell wall anywhere. We will spend a fair amount of time talking about most of the bacteria. Now, the blue-green algae are the photosynthetic prokaryotic cells. They're interesting, they're important environmentally, but they're not important from a pathogenic standpoint because they make their own food. The eubacteria, the chlamydia, and the rickettsias do have species that cause disease, and we'll talk more about them. The archaea are methane producers. They're halophiles, which means they can live in high salt environments. They're thermophiles. They can live in very high heat environments. They too are interesting from a standpoint of the environment. We will not talk again about the archaea, but they are pretty. This is a hot spring at Yellowstone National Park, and the lovely orange coloration you see here are some of those archaea, those cells that are prokaryotic cells. They are living in the hot, acid environment of this volcanic spring. Just to give you a little perspective, here are 2 large eukaryotic cells. with their nucleus. And here are some prokaryotic bacterial cells. Prokaryotic cells are typically much, much smaller than eukaryotic cells. The kingdom fungi, which would be in the domain Eukarya, are all composed of eukaryotic cells, the membrane-bound nucleus, the multiple organelles. These are the molds and the yeast, and some of these are involved in disease. A lot of these are involved in food production. Now, molds are the fuzzy things, and yeast are more like a cell, a eukaryotic cell. If you've ever had green little fuzzy stuff grow on your oranges in your refrigerator, that was a mold. If you have ever smelled that wonderful smell of bread baking, that's the smell of yeast. The kingdom Protista, also in the domain Eukarya, so eukaryotic cells. We have the protozoa, and protozoa are classified by means of their motility. So they move with pseudopods, cilia, flagella, and some of them have no organelle for motility, and that puts them in a class by themselves. So here we see an organism with the pseudopods, little false feet they stick out and flow to. Here is one with cilia. Cilia tend to cover the surface of the cell and are used in motility. And then here is an organism with flagella. Now also in the kingdom Protista are the algae. Algae again are eukaryotic cells. These contain chlorophyll. Chlorophyll means that they're photosynthetic, and if they're photosynthetic, they make their own food, so they don't have to live at the expense of another organism. These are not going to be involved in disease, but they are important environmentally. The algae give classificationists a little bit of a problem, because algae are everything from single-celled organisms all the way up to some of that giant kelp and seaweed that you may have seen on the beach. The people in classification are like, are they really in the Protista kingdom, because that's more like single-celled things, or small multicellular things, or should they be plants, because that kelp and seaweed is pretty good-sized stuff. So we're not going to worry about that. Just know that algae are photosynthetic. Now here you see Spirogyra with their green strands of chlorophyll. And these lovely things over here are called diatoms. Diatoms, in addition to having chlorophyll and being able to make their own food, have these unique shapes because they have cell walls. And their cell walls will have in them things like calcium or silica. When the organisms die, they will fall out as sediment at the bottom of a body of water, and this stuff will be harvested and called diatomaceous earth. And these little tiny cellular things here, the little leftover cell walls of these things, make excellent polishes. So you find diatomaceous earth in a lot of polishes for silver and fine jewelry, and even for your teeth. Now the kingdom Animalia is where we find the helminths, and the helminths are the parasitic worms. They are all eukaryotic. We study them in microbiology because we have to identify them by looking at their eggs, which are microscopic, or at their larval forms. But they are pretty good sized worms for the most part. Now this is a worm in the bloodstream. This is the Loa worm. This is a red blood cell here. Here's a white blood cell here. So you can see it's pretty small. This is a parasite that is seen in West Africa. The worm can travel in the blood, and then it will get out and wiggle through the tissues. It's usually not too terribly irritating for the person that has it, except when it is migrating across the conjunctiva of the eye. That gets a little irritating. West African natives who have this are not noticeably ill, but they do have inflammation and a few problems with their immune system as a result of this infection. We also are going to study viruses in microbiology, but viruses are not really living. They only display the characteristics of life if they have invaded a living cell and sort of hijacked its RNA, DNA, and cellular organelles. As a result, they're not classified in any of the Linnaean classification system. Viruses have their own separate classification system that talks about whether they have DNA or RNA and how they replicate and some things like that we'll go into later. Now this is a virus. The pink things are viruses. On the outside, these things that look like glorified lunar landing modules are actually the virus attaching to the outside of the cell. they will inject, in this case, their DNA into the cell and take over this host cell's whole metabolic processes and force the host cell to make many, many copies of the virus. When enough copies of the virus have been made, the cell will rupture, thus killing the cell, and all of these new virus particles will be out invading other cells. Now that gets us through classification, and that's about all we're going to say about classification. But now we're ready to talk about what happened after Leeuwenhoek and after Linnaeus. We enter a period about 200 years after Leeuwenhoek called the Golden Age of Microbiology, and it's in the mid to late 1800s. It's about 50 years when there were just all kinds of concepts and theories developed about microbiology. While microbiology itself is a sub-branch or a branch of biology, there are many branches of microbiology, and most of these were developed during this golden age of microbiology. One of the things that developed early in the golden age of microbiology was the development of scientific method. And this was simply a standardized approach to answering a question about something in nature. Scientific method starts with somebody observing something and then generating a hypothesis. It's kind of like, I wonder why this happens. Then generating a hypothesis like, well, I think it happens because of this. You then have to test this hypothesis with experimentation, and you've got to be careful about your experiment and make sure you set it up so that the only thing that varies is the thing you're really looking at and everything else is held constant. And as a result of your experimentation, you either accept your hypothesis, you reject your hypothesis, or you may modify your hypothesis and retest. Any of those can happen. So if we start with observations, sort of a question, why does this happen? And the hypothesis is really an answer or a possible answer to that question. You develop an experiment, and it says here including control groups. You may have heard of experiments where someone was trying to determine if a particular drug was effective against something, and you heard that one group got the drug and the other group got a placebo. That is, if the group getting the drug were getting pink pills, the control group had to get pink pills that were sugar pills. They had nothing in them, just basic inert stuff. Because some people will think they're getting better just because they think they're getting a treatment. It's called the placebo effect. So you've always got to have those control groups that have the same thing done to them, sort of, kind of, but not the actual experiment done. After you have conducted your experiment and made observations, you may find that your experimental data does not support your hypothesis. It just didn't work out. At that point, you have one of two things you can do. You can just reject the hypothesis. I was just totally wrong, forget this. Or you may say, hey, I think maybe I didn't take into consideration something. You modify your hypothesis and you go back, modify the experiment, and go through the experiment again. If your observations lead to data that supports your hypothesis, you can accept your hypothesis. Typically, one scientist accepting their hypothesis is not enough. Other scientists will continue to test that hypothesis and continue to support it. Every once in a while, some scientists will come up with an experiment and we'll get data that does not support the hypothesis. Then we have to look carefully at their experiment. We have to determine whether they were really testing what they said they were testing. But eventually, a hypothesis becomes proven enough times that it becomes a theory. So we talk about theories. These are things that have been tested repeatedly, and every experiment has supported the hypothesis. Now, if things just become like incontrovertible, there is no way this does not work this way, it becomes a law. So we have the theory of relativity, but we have the law of gravity. Everybody's pretty sure that gravity thing works no matter what. A big question that went on as the golden age of microbiology is starting is the controversy over biogenesis versus spontaneous generation, or what is called abiogenesis. Biogenesis means life comes only from living things. Abiogenesis, or spontaneous generation, was the statement that, oh no, things just spring up from anything. This idea of life springing from non-living things actually came from Aristotle, and so he stood as a pillar of belief for a long time. Who wants to cross Aristotle? His basic tenet was, well, if you leave meat out, it eventually gets maggots in it, and maggots are living things, so that dead meat gave rise to life, maggot. Long about the beginning of this age of the golden age of microbiology, there was an Italian scientist named Reedy who said, I'm not so sure about that. I kind of see that flies crawl all over that meat before the maggots come. Now maggots, which are fly larvae, look very different than flies, so a lot of people didn't make that connection that maggots turn into adult flies. But Reedy kind of made that connection. He developed an experiment where he kept the meat isolated from flies. He had meat in an open flask, flies got to it, sure enough, maggots grew. But if he sealed the flask, no flies, no maggots. If he even just covered the flask with gauze so flies couldn't get to it, no maggots. So this began the first little chink in the armor of spontaneous generation. But it wasn't over yet. A scientist named Needham boiled beef infusions. Now beef infusions are very nutrient rich and will grow bacteria in a New York minute. And he sealed these with corks, and in just a few days' time they got cloudy. And he took this to mean that no, non-living things have a life force in them that's going to grow stuff. So he swung over to the Abiogenesis group. Spallanzani was not happy with that. He said, I am not so sure about that. He heated his boiled beef infusions, he heated them longer, and he sealed the glass vials. Now the criticism here was that the prolonged heating destroyed the life force, and when he sealed the glass vials, he also eliminated vital force from the broth. Vital force is something we today call oxygen, so he was not letting it there. So there was no air to support life. Well, along comes Louis Pasteur. And Louis Pasteur was one smart cookie, let me tell you. He developed a biogenesis experiment that pretty much laid to rest the whole thing here. He developed a special swan-necked flask so that he could put his infusion into the flask and boil it, and then he left it open so that air could come and go. Okay, now there's plenty of air. As air moves in and out of the flask, what happens is dust from the air will settle in here. That dust is where the microbes are. But microbes can't crawl. They can swim, but they can't crawl. So as long as this stayed nice and dry, none of those microbes got over here into his beef broth, and the infusion would remain uncloudy for months. Absolute months. You would think it would not, but it does. Now Pasteur noticed that if he took his flask and he tilted it so some of the broth flowed down into this part of the flask, and then set it back up, in a day or two it would be cloudy. So he said, just because we can't see them doesn't mean there aren't living things in the air that is responsible for life. This pretty much rejected the spontaneous generation theory and greatly supported the biogenesis theory. Big wine crisis in France. France is noted for its fine wines. And suddenly, the wine in France is not alcohol, but vinegar. Oh my goodness, this is terrible. So the vintners of France put up a big reward for anyone who could figure out what was going on. Pasteur took the challenge, and by the way, won the challenge. He decided to use this as yet another experiment to sort of support biogenesis and disprove abiogenesis. So he started out with his observations. When grape juice fermented, if he looked at it microscopically, see, we had microscopes back by then, he saw yeast cells and bacterial cells. So he came up with 4 different hypotheses. The first hypothesis was fermentation is spontaneous. This would be, abiogenesis. He heats a flask to kill all the microbes. He seals it, and all he gets grape juice. Nothing happens. So we reject this hypothesis. Fermentation is not spontaneous. Well, maybe it's air that ferments grape juice. So he boiled some grape juice in his special little swan neck flask, left it open to the air, and no fermentation occurred. Reject hypothesis number 2. Third hypothesis. We had bacteria in that grape juice before. Let's see if bacteria ferment grape juice into alcohol. Boils the flask, inoculates it with bacteria, seals the flask, waits a while, and bacteria do something to the grape juice, but they make acids, and acids are vinegar. This does not taste good. This is the problem with the wine. So Pasteur moved on to theory 4, hypothesis 4, that the yeast were the ones that fermented the grape juice into alcohol. Again, boiled the grape juice, added the yeast, and yes, lo and behold, yeast produced lovely alcohol. We now accept the hypothesis that it is the yeast in the grape juice that is responsible for developing the alcohol. Now, Pasteur used this to develop what's called the germ theory of fermentation, which is simply that different organisms metabolize the same substrate, in this case grape sugar, to different end products. Bacteria give you acids, yeast give you alcohol. Now, we think of fermentation a little differently today. In Pasteur's time, the term fermentation sort of meant the same thing as metabolism. Basically, that these organisms would use the grape sugar to produce either acid or alcohol. Now, Pasteur also came up with a little thing called pasteurization, which was not originally made for milk, but was made for wine. And pasteurization is simply heating a fluid to a high enough temperature to remove unwanted organisms, but not remove desired organisms. In the case of the grape juice, we wanted to kill the bacteria, but leave the alcohol-producing yeast behind. Pasteurization has now been applied to a number of fluids, such as milk, to heat it to high enough temperature to remove pathogens from milk. Pasteurized milk is not sterile unless it's ultra-pasteurized milk. Pasteurization is also used on some beers to stop the fermenting process by killing the yeast. But this began the industrial microbiology and the era of biotechnology. When we talk about biotechnology now, we think of real high-class, ritzy kinds of experiments with gene splicing and things like that. But biotechnology is really having an organism do anything for you that makes your life better. So we've been using biotechnology for centuries. We've been making bread and wine and beer and ale and all kinds of things using yeast and bacteria for centuries. We've just gone a little higher tech with it recently. Pasteur also developed the germ theory of disease. He began to believe that specific microorganisms, unseen, could cause disease. And this was a pretty big step. A lot of scientists did not believe this at the time. Pasteur also took on some of the work of Jenner. Jenner created the first vaccination for smallpox using cowpox from cows. Pasteur continued to work with his ideas. He worked with fowl cholera and anthrax and rabies to develop vaccines for these. Now, the vaccine for rabies is pretty interesting. Rabies was a pretty common disease in Pasteur's time, and rabies was a certain death sentence. Pasteur had been working for a long time on a possible immunization that would stop rabies from killing an individual. One day, he hadn't been able to use it on anybody because, well, it was untried, and you don't really want to try it out on somebody and have them die as a result of it. One day a mother came to Pasteur and begged him to use his new vaccine on her son. He had been bitten by a rabid dog. Pasteur looked at her and said, "But it may kill him." The mother looked at Pasteur and said, "He's going to die anyway. Why not give him a chance?" Pasteur began injecting the boy with his vaccine. A week passed, no change. 2 weeks passed, no change. 3 weeks passed. At the end of 3 weeks, the boy began to get better. He went on to recover. He was a farmer for many years, and when he retired from farming, he moved to Paris and was a doorman at the Pasteur Institute until the mid-1940s when the Germans came into Paris. As a result of his work with immunizations, Pasteur is sometimes called the father of immunology. Now here are just some of those industrial uses of microbes. Some things you may not think of. See, there's that diatomaceous earth. Drain openers, pest control chemicals, the more common things you may have known about, bread and sour cream and cheeses, and of course alcoholic beverages. Now, another scientist of the time, Robert Koch, was a German, and he is credited with developing or starting the science of etiology. Etiology is the cause of disease. He had heard of Pasteur's germ theory of disease, that one organism caused a specific disease, and he started out trying to prove that. He worked with anthrax and as a result developed Koch's postulates. Now, Koch's postulates go like this. You isolate an organism in every case of the disease you're studying. You grow that outside of the host in pure culture. Then you take that organism, introduce it to a healthy subject, and cause the same disease. You then re-isolate that same organism from that second host. Now, this is still the gold standard used today to show that an organism causes disease. There are some exceptions. We have some organisms that do not grow outside their host, like the organism that causes syphilis, treponema, or the organism that causes leprosy, Mycobacterium leprae. We have some diseases that humans are the only known host. That makes giving a disease to that second host a little unethical. So there are some problems, there are some workarounds we do, but we still use this. Koch, because of the work he did, he made a great many advancements in microbiology. He developed petri dishes. He found agar as a solidifying agent. He worked with simple stains. He was the one who came up with a way to transfer bacteria aseptically from one place to another using sterile loops and needles. And he came up with a rough way to enumerate cells. So here you see a lot of different looking colonies here, and as long as we can get them all by themselves, we call them isolated colonies, and we assume that they all came from a single cell, so they're all the same thing, they're pure cultures. Now, there were even more advances in microbiology. Hans Christian Gram developed the Gram stain. This is a differential stain that is used in identifying bacteria. It's the cornerstone of microbiology today. It's the first thing you need to know about an organism to start working with it. Ignaz Semmelweis was an Austrian physician, and he came up with the whole concept of hand washing. You'd have thought hand washing was kind of a given, but it wasn't in those days. Semmelweis worked at an Austrian hospital where there were 2 maternity wards. One maternity ward was run by midwives. The other maternity ward was run by physicians. Now, the death rate on the physicians' maternity ward was so high that women would beg not to be placed there. The system was, one day you went to the doctor's side, the next day you went to the midwife's side, then the doctor's side, back and forth. You had women giving birth in the street rather than go to the doctor's side if that's when they went into labor. Semmelweis did some observations, that good old scientific method thing going again. He noticed that when he walked into the doctor's side, it was dirty, it was filthy, there was a bad smell. He noticed that the physicians would have come straight from autopsies into the maternity ward and examined their patients. When he went to the midwife's side, he was immediately struck by the cleanliness. Clean linens. The midwives changed their aprons when they got dirty, and they were constantly washing their hands. Now, Semmelweis wanted the physicians to just wash their hands when they came on duty. He picked a lime chlorate kind of thing, something that was used in the autopsy room to help cut the smell. It's a little hard on the hands, so the physicians weren't too crazy about using it, but for a little while they did wash their hands in that. And while they were doing that, the death rate on the doctor's side went down. It never got as good as the midwife's side, but it got much better. So Semmelweis, sort of spread the word that hand washing might be a good thing to do. Again, it was not readily accepted. The story goes that Semmelweis went a little crazy because the physicians stopped washing their hands and the death rate went back up, and he couldn't explain it. He didn't know they'd stopped washing their hands. He was committed to an insane asylum and died a few years later. He died relatively young from childbed fever, the very thing that the women died from in the maternity wards. It is said that one day he escaped his bonds, and where his wrists were cut and broken from being tied up all the time, he had open wounds. He went to the autopsy room, plunged his hands into the body of a woman who had died of childbed fever and died of childbed fever himself a few days later. That's kind of an over-dramatization of his death. He was committed to an insane asylum. He did die of an infection that he contracted while he was there. I don't think he really went plunging around in dead bodies, but it's such a good story. Now, Joseph Lister was working in London, and he was aware of Pasteur's theory about disease and Semmelweis's success with hand washing, and he thought, gee, I wonder what would happen if we disinfected the surgical instruments in between patients. I know, gasp, you would think they would have figured this out, right? But they didn't. At the very best, they would throw all of the tools into a bucket of bloody water and then yank out the tool they wanted for the next patient. Patients were literally just hauled off and on the table. Nothing much happened. So Lister began using carbolic acid, which was an antiseptic of the day, to soak the instruments in, and he had people wash their hands in it before they did surgery, and he would scrub the wound area of the patient they were going to work on, the area they were going to do surgery on, to try to remove bacteria from the skin. And his death rate from surgical infections went down dramatically when he did this. Unfortunately, carbolic acid is a little hard on the skin, and so it sort of interfered with wound healing a little bit, but no more death from sepsis. Florence Nightingale is credited for bringing cleanliness into nursing. Now, Florence Nightingale was born in Florence, Italy. That's why she got the name Florence. But her family moved to England when she was a year old. She was born into a wealthy family. It was expected that in the 1800s she would grow up, marry well, have children, be a housewife. Florence wanted none of this. She was a little bit of a rebel in many ways. She did not like the idea of a life of being idle. She got training as a nurse, and during the Crimean War in 1860s, she and a small group of similarly trained people that she had trained, went to Crimea to nurse the soldiers. At the time she got there, about 40% of the deaths were from infections, not from the wounds themselves, but from the infections, because there was just no standard of cleanliness. After she and her nurses had been there for a while and raised the standard of cleanliness, the death rate went down to about 2% from infection. They still died of their wounds quite frequently, but at least it was not infection. Florence Nightingale was known as the Lady with the Lamp because she was known to make rounds among the patients even late at night when everyone else had gone to bed. Florence Nightingale was not without her distractors. It was not all easy for her, because she was a woman and she went into a war zone area to treat men. Up until that time, the only women that followed men into war were called camp followers. And yes, they did get paid for doing laundry and cooking and other services that were provided to the men by them. Basically, they were prostitutes, and there was a small contingent of people that wanted to make Florence Nightingale and her nurses out as prostitutes, so she did have her distractors. But overall, she developed the first real school of nursing at St. Thomas Hospital in London, and she brought the concept of cleanliness into nursing. John Snow is credited with developing the science of epidemiology and infection control. Epidemiology is finding the source of a disease. In London, in the late 1800s, there was a severe outbreak of cholera. John Snow, Dr. Snow, tracked down where the people were getting the cholera from. He found there was one infected well in London, and once he got that well shut down and got pretty much everybody well, they either died or got better, he stopped the outbreak of cholera. So he is credited with the science of epidemiology. Edward Jenner, as I mentioned earlier, made the first vaccine. He noticed that the milkmaids of England had beautiful fair complexions. They were not marked by smallpox pox. He decided that the reason was, he noticed that when they milked cows, cows would have cowpox, that they would sometimes get furious blisters on their hands from milking the infected cows, but that this seemed to give them protection against smallpox. So he developed a system of introducing cowpox into people, which didn't cause smallpox in them, but gave them immunity. Even before Jenner, it was a fairly common practice to get material from smallpox pustules of someone who was dying of smallpox and inoculate that into healthy people to try to provide protection for them. Paul Ehrlich is credited with chemotherapy, developing chemotherapy. Dr. Ehrlich was looking for what he called the magic bullet. He was looking for just the right amount of a toxic chemical that would kill the disease-producing organism without killing the host. And what he was going after was syphilis. Now, he used arsenic, and he tried varying amounts of arsenic until he got just the right amount of arsenic that killed Treponema pallidum without killing the patient. So our first chemotherapeutic drug was arsenic. He called it compound 606 because the first 605 didn't work well. And it also got the name of salvarsan. And this came out in, like the 1920s or so. Fleming and Domack were both pharmacologists. They were both doctors who came up with other substances used to treat infection. Fleming came up with penicillin, which is from a fungus. Quite by accident, he noticed a fungus growing on one of his bacterial plates. A Carolus lab assistant had left the lid off the plate, and he was getting ready to discard it when all of a sudden he noticed that, whoa, bacteria are growing around that fungus. And he began exploring why that happened, and he began to harvest penicillin from the mold. Domack came up with sulfa drugs. Now, Domack won the Nobel Peace Prize in 1936. He was German, and he was not allowed to leave Germany to collect his prize because Hitler was not letting his scientists leave Germany at that time for fear that they might not come back. As we move into the modern age of microbiology, that is the 19th century, we have Cliver and Neal who developed the concept of biochemistry. They discovered that all living things share the same basic biochemical reactions. They also showed that energy is based on the transfer of electrons from one chemical to another. So that oxidation reduction stuff that you all hate so much in chemistry, blame these guys. Also, we began exploring microbial genetics. There were 3 scientists, Avery, McLeod, and McCarty, who conducted an experiment and didn't really know what they had done. They were looking for a vaccine for Streptococcus pneumoniae, which causes bacterial pneumonia. Now, there are 2 strains of Streptococcus pneumoniae. One is what is called the smooth strain. It has a capsule around it. And then we have the rough strain, which does not have the capsule around it. Now, when you put the smooth strain in mice, it kills them. If you put the rough strain in mice, they survive. So with all of the different things that Pasteur had done and Jenner had done about, killing the pathogenic strain and putting it in mice, the mouse survived, but they had no immunity. So they tried using the live strain that did not kill mice, as well as some of this heat-killed strain. Now, if you look at it, the rough stuff doesn't kill the mouse. The killed smooth stuff doesn't kill the mouse. And yet, when they gave the mouse both smooth and rough, the mouse died. Upon mouse autopsy, they recovered some living smooth strain. They said something inside this dead stuff transformed the living stuff. It was another 30 years or so before they found out that that something was DNA. But this was the first step that showed there is something in cells that controls how cells look and how they behave. DNA is what controls an organism's characteristics, and genes are contained in DNA. It was Beadle and Tatum, about 30 years after the experiment we just talked about, that came up with the concept that one gene codes for one protein. And proteins, all of the chemical reactions in your body, are controlled by enzymes, and enzymes are proteins. So one gene controls one biochemical reaction in your body. Linus Pauling began gene sequencing and the whole Human Genome Project. And as a result of all of this, we developed recombinant DNA and gene therapy, where we learned to splice out genes and move them from one organism to another so that we were able to get bacteria that could make human growth hormone. We have bacteria that can make factor VIII for hemophiliacs, for example. And now we're doing more and more studies where we find a human that has a defective gene, and we're clipping out the defective gene and putting in a not defective gene. And if we can do that in enough cells and get those cells to multiply, we may take care of some diseases like cystic fibrosis. We know that the body can defend itself. And again, in the mid-1800s, there was a discovery that there were certain blood cells and certain chemicals in the blood that seemed to fight disease. This gave rise to the sciences of serology and immunology. Couple that with the chemotherapeutic agents and antibiotics, and add hand washing, and you have a pretty strong way to prevent people from getting sick. It is said that in the 1900s, in the 20th century, the development of antibiotics and chemotherapeutic agents were the one thing that lengthened life, the thing that lengthened life the most. You're going to hear me talk about hand washing a lot. It is the easiest, cheapest, most effective way to prevent the spread of disease. So what does the future hold for us? Well, perhaps we can eradicate certain diseases like tuberculosis. We almost had that gone when HIV reared its ugly head, and a lot of research money went to that. So maybe we can get things like that. Smallpox has almost been eradicated. There are several diseases that we would like to see go away. We have a lot of things building up in our landfill because we don't have anything that naturally degrades that. Maybe we can genetically construct some microorganisms that can eat styrofoam and start bioremediation. We do know that organisms develop a resistance to certain drugs, so in the future we need to look at ways we can defend against those drug-resistant organisms. And then we have new diseases appearing all the time. These diseases that have occurred in the last 20 years or so, or maybe have been gone for a while and are coming back, are called emerging diseases. One emerging disease is something called the variant Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease was a genetic variant. It was seen in a small group of people, and these people have for some reason develop neurofibrillar tangles in their brain that causes their brain to short circuit. Their brain gets very spongy and eats out in big holes, and they become unable to function appropriately. In the genetic form of the disease, it takes several years for the full-blown disease to develop, and it's seen only in older people. There were a couple of other spongiform encephalopathies besides Creutzfeldt-Jakob that were out there. There was a disease called Kuru, which was seen in certain Polynesian natives that practiced cannibalism, and there was a disease called scrapie in sheep, where the sheep, over long periods of time, would develop an inability to stand up straight. They would stagger around, and when you would look at their brains had big holes in them. They were spongiform, and they did not look like they should look. The cannibalized natives, turns out they didn't eat just any part of somebody. Eating the brain of a dead family member was considered a way of mourning. And so they would see that if you were a close friend, you might also be invited to eat the brain of your friend that had died. Eating the brain seemed to be the thing that caused the Creutzfeldt-Jakob disease to develop. Well, about 15 years or 20 years ago, there was an outbreak of scrapie in Wales, and the sheep had to be destroyed. Animals need protein, and you don't often see them ordering a burger someplace, so protein is supplemented into the grain feed that they are given. They took the sheep carcasses from the diseased sheep, ground them up, and added it to cow food. It was the protein in cow food. And in a few years we began seeing a disease called mad cow disease in Europe, particularly in England, because that's where most of the feed went. Then after those cows had been in the food chain for a while, we began seeing this variant Creutzfeldt-Jakob disease in, unfortunately, relatively young people, and when they get the variant form of the disease, it is very rapid moving. If someone lasts 3 years with this, they've lasted a long time. And again, it destroys the brain. Now, we believe the causative agent is a small protein particle called a prion, and it's resistant to any kind of disinfecting or autoclaving that we've been able to do. But we think that somehow this prion got into our food chain, and this is what is causing the variant Creutzfeldt-Jakob in Europe. But that's a case of an emerging disease.